Hello, everybody. I thought I would take a little time here and do a little bit of review before the Unit 1 exam, mainly focusing in here on the physiology exam more so than the anatomy side for your Biology 201 Unit 1 exams. Let me go inside our class here and just remind you about a couple of things that you should be doing to help you prepare for the test. Let me switch over to the student view here so it looks like what you guys are going to see rather than what I'm going to see. And if I go back into Unit 1 over here, come on Blackboard, we're being slow. Here we go. Okay, and again, if you have not already done so, go through your tasks for Unit 1 because that's going to have a lot of pointers for how you should approach the class. Unit 1 Anatomy folder here, that's where all your anatomy content, all your content for the Unit 1 anatomy test is in here. Open this module, be sure you've gone through, there's the study guide here, be sure you've got that printed out and you know everything that's listed on the study guide, how to identify it, all of the terms there. Be sure you refer to the textbook pages and so forth that are mentioned. Like be able to identify and name all of the body regions in figure 1.7 on page 13 of your textbook. Be sure you practice with all of those things. It's a lot of lots of terminology. Once you've been through that module, there are some practice worksheets that are located here for you guys to go through, and the answer keys are there, so work on them and then check your answers against the answer key. If you have any questions, please put those on the Piazza discussion board so we can talk about them. And then please don't forget this, the little L assignments. That's your anatomy review. So as you click through there, you've got lots of questions that are not going to be identical, but there are activities and questions that are going to be very similar to what you're going to see on the test. So like right here, this picture of the brain, that's not going to be identical to what you're going to see on the exam, but it's going to be very similar, and you just type in your answers here in the blanks. And the only thing you have to be careful about is spelling. This activity is going to really help you practice with your spelling, because if you misspell a word, then it marks it wrong. So it's a great way. You can keep doing these over and over and over again. They're not for a grade, and it's a great way for you to practice your spelling as well as your identification of these anatomical features. So your mastering A&P homework, of course, that is for a grade. These L activities that you see are strictly for practice, and you can do those over and over again, but I can't emphasize enough how much you want to use those to help you practice. That's what they're there for. I spend a lot of time making them, so use them. All right, Unit 1 Physiology inside this folder. Again, you got a study guide for the Unit 1 Physiology exam. Be sure you have printed that out and you've gone through everything. It's not a study guide. You guys who have not been out of high school for very long, it's not going to be a high school style study guide. Memorize and regurgitate. Memorize these fill in the blanks or whatever, and you've got the test. It's just going to provide some pointers about things that you need to review and think about while you're studying. Be sure you've gone through Chapter 1, Chapter 2, Chapter 3, those modules. Click on those, open them up, go through the material, the video lectures in there, and then you also have an L activity for the physiology side. So if you open that up, this is not for a grade like your mastering A&P homework, but you can go through here and just like with the anatomy side, lots and lots and lots of practice questions for you to go through. Also, you can go back to your mastering A&P homework even after you have completed the homework for a grade. You are supposed to be able to go back and revisit those questions as many times as you want, not for credit, but just for practice. The more you practice with actual questions, that's going to make you think about the material and test your knowledge. And that's the thing. After you've reviewed and studied, that's the key thing that you need to do to help you prepare for these exams. Okay, so I just wanted to kind of remind you of those things. I've got a few practice questions here that we can go through, though. I have used these in my traditional classes. And it may help if we just kind of talk through some of these things. So we'll go ahead and run through these example questions, and I will post these on blackboard for you. Let me bring up a pen here so that I can actually write on here. And I got to change colors. Okay, there we go. All right, how about this first question? All right, so I'm going to go through the questions and the answers, but you can take advantage of being able to pause YouTube. So instead of just watching kind of passively and listening to me go through these questions and the answers, you can pause and see if you can answer the question yourself before we get to the actual answer and the explanation. So use this wisely as well. How about this first one? Which of the following is not a life requirement for humans? So part of the very first section of chapter 1 describes some of the basic requirements for life. So which of these would not be a life requirement? We've got oxygen gas, pH 7 for body fluids, chemical nutrients, normal atmospheric pressure. All right, so what's your guess on this one? I think we all know that oxygen gas is required for life, so we can eliminate that one because it says not a life requirement. That's one thing with testing. Slow down. A lot of students speed through exams. They're not reading carefully, and that can be their downfall on tests. So give yourself plenty of time. I know a lot of you guys have major life constraints, but try not to get to the testing center and wind up with barely enough time to take your test, because you need to slow down and read carefully. You got a lot of new terminology that you've never seen before. This is almost like taking a foreign language class at times, so you've got to be able to slow down and read carefully. pH 7 for body fluids. How about that? That might sound kind of reasonable. Chemical nutrients. Yes, chemical nutrients are a life requirement. Okay, things like glucose, sugar, those are chemical nutrients. We absolutely need those to survive. Normal atmospheric pressure, we can rule that out. If we didn't have normal atmospheric pressure, we could not survive. If you've ever flown in an airplane, you know they talk about they're going to have supplemental oxygen come down if the cabin depressurizes and the pressure is too low for normal breathing to take place. So the answer here is pH 7 for body fluids, and the reason for that, hopefully you guys remember, normal pH range for our body fluids is what? 7.35 to 7.45. That's one of those things you want to start learning right away because that comes up fairly frequently. How about this next one, body systems? We had an overview of the various body systems that you're going to be studying in more detail in biology 201 and 202, so you want to go back over those basic functions. So this question says, which body system is responsible for gas exchange between the blood and the air? Is it cardiovascular, urinary, digestive, or respiratory? I'll let you guys pause. Okay, carrying on. Gas exchange, think respiratory. This is the function of the lungs in moving oxygen gas and CO2 gas back and forth between the air and your blood. The cardiovascular system transports gases and nutrients to all parts of the body, so it's more involved in transportation. You have these blood vessels extending all throughout your body, so that's how we ruled that one out. Digestive is not responsible for gas exchange. Digestive is responsible for breaking down the foods that you eat and absorbing those chemical nutrients. Urinary system filters the blood and eliminates wastes out of the body, so it's not going to be functioning in the gas exchange between the blood and the air. So that left us with the respiratory system as our answer. All right, how about this question? Select the two body systems most associated with controlling other parts of the body, either short or long-term control. There are two body systems that you should start thinking about early as being involved in controlling your body. So we'll go ahead and pause for a second to let you guys try to figure this one out. Okay, what are our answers? A and E are the answers, the nervous system and the endocrine system. And you guys know the brain part of the nervous system involved in control. So that one's pretty easy. And so the other one you just have to remember is endocrine. The endocrine system is responsible for making hormones. And you guys know that hormones are very important for various bodily functions and proper development of the body and so forth. And so, just get used to thinking of hormones as these chemicals that are also involved generally in longer term control of our body processes. Okay, so those are those two things. And we've already talked about the key things with cardiovascular: transporting substances throughout the body; digestive, taking in, breaking down and taking in nutrients; reproductive is involved in that gas exchange between blood and air. All right, here's a homeostasis question. Homeostasis is a really important topic for Unit 1. Which of the following is not an example, keyword there, not, of negative feedback regulation? All right, so let's just go through these and see if we can figure it out. I'll pause, let you pick the answer. Okay, and now let's go back through. How about this one? High body temperature triggering sweating and dilation of superficial blood vessels to release body heat. Is that an example of negative feedback? Remember, negative feedback involves bodily processes, levels of substances like sugar and hormones and so forth that have to exist within some normal range. They tend to fluctuate. They go up, and then there's a mechanism that kicks in to cause the levels to go back down, and if it drops too low, there's a mechanism that kicks in to cause it to go back up. That's negative feedback regulation. You tend to reverse the trends of processes. So A, to me, sweating, dilation of blood vessels, that occurs when your body's getting overheated, right? And the idea there is to cool your body temperature off. So that is negative feedback. B, dehydration triggering the thirst response that you will consume, so that you will consume more water. So you're getting dehydrated. Body water levels are dropping. You get thirsty. You consume more water. Water levels in your body go back up. So that looks like negative feedback to me as well, because we are reversing a trend. Contractions of the uterus becoming more and more forceful until a baby is delivered. That's positive feedback. All right, so that's our answer right there. Uterus starts contracting when a baby's going to be delivered. That sends signals for release of oxytocin, that a hormone called oxytocin. That hormone travels back to the uterus and triggers the uterus to contract even more forcefully until you actually squeeze out the baby. So there, the trend is not being reversed. The trend is for greater and greater and greater and greater contractions until the baby is delivered. D, low blood sugar levels triggering the liver to break down glycogen into glucose monomers or building blocks, which are released into the bloodstream. That's going to be, you've got a case where your blood sugar levels are dropping. So the liver stores glucose in chains in the form of glycogen, chains of glucose molecules. And so if you have low blood sugar, then the liver breaks the glycogen down into individual glucoses, releases those into the bloodstream. That causes your blood sugar to go back up. So that is an example of negative feedback as well. Now I know in that case your blood glucose is going from a lower level to a higher level, so that almost might sound like it should be positive feedback because your blood glucose is going to go up. But that's not what negative feedback means. Negative feedback means that you're just reversing the current trend. So if blood sugar levels are dropping, you're sending them back upward, as opposed to just building on the current trend like you have with a uterus contraction. Contraction gets stronger, 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 stronger. That's a positive feedback example. Okay. Don't forget to review all of the different steps of homeostasis, where you have your receptor that detects a stimulus of some type, and that sends a signal to a control center of some type. The control center sends a message out to effectors that are going to make a response that's going to reverse a trend, like with body temperature. And so you've seen this diagram in your textbook, so just be sure you review that textbook and diagram and kind of understand all of the different parts of a typical series of things that happens with homeostatic control. All right, how about this example question? When you take Biology 202, you're going to learn that high blood sugar levels trigger cells in the pancreas to release a hormone called insulin. Insulin stimulates many cells in the body to take up glucose, which lowers your blood sugar levels. What is the response in this scenario? What is the response? The response is something that happens at the end of one of these homeostatic control mechanisms that we're learning about. What's the response? So let's just go through the answers. The response is going to be the thing, how your body is changing in adjustment. What response is your body making to reverse a trend? And here we have high blood sugar levels. Blood sugar levels are going up. Okay, so is it high blood sugar levels? Is it release of insulin hormone? Is it lowering of the blood glucose levels? Or is it detection of the high blood sugar levels? All right, so I think it's pretty easy to rule out A and D. So the question is it release of the insulin hormone that's the response, or is it the lowering of the blood glucose levels? All right, the response by your body is actually the lowering of the blood glucose levels. This is the thing that's going to happen at the end, where the trend that we're trying to reverse is actually being reversed. So our trend here was increasing blood sugar, so our response needs to be lowering of the blood glucose or blood sugar levels. The release of the insulin hormone here is going to function like your effector, because it's going to be the thing that causes the response to occur. Once the pancreas releases insulin, insulin tells cells in your body to soak in all this glucose that's in your blood and body fluids, and that leads to our response, which is the lowering of the blood glucose levels. All right, how about this one? Some basic chemistry stuff here. And again, with the chemistry, a lot of it is just kind of getting a mental picture and practicing with the terminology. Don't try to get too stressed out over it. Freak yourself out over the terminology. So with chemistry, we learned about inorganic chemicals. We learned about organic chemicals. Just keep in mind the organic molecules are carbon-based, and most of them fall into those 4 major organic macromolecule categories. Don't forget about those. Be sure you study those and what they're built from. So which of these would we classify as an inorganic substance? So this is one of those where more than one is correct. So you'll read really carefully with the questions to make sure you know whether there's just going to be one answer or whether you got to select more than one. So let's just go through these. Water, is it organic or inorganic? Water is H2O. So we just said carbon, organic compounds are carbon-based. So that's a big clue right there. That's inorganic. Carbon dioxide. Now I know carbon dioxide is CO2, and so you might think it's an organic compound, but it's actually not. It's a little small molecule, and it also is part of the inorganic category. Proteins. Proteins are one of the 4 types of organic macromolecules, so it's going to be organic. Monosaccharides or sugars, that's another name for sugars. They're definitely in the organic category. A sodium ion, that's just a sodium atom that has a positive charge to it. It's lost an electron. And so ions are not generally classified as organic molecules. Now there are some types that are organic ions, but most of the ions that we're learning about in this class, like sodium ions and calcium ions and potassium. Those are the ones you're going to hear about the most in your 201 and 202 classes. Those are in the inorganic category. All right, how about acids and bases? Here, review the basic information about pH. A solution that contains a very high concentration of hydrogen ions would be considered which of these? This can be a little bit tricky, but so let's review. Remember, if you're, all right, if I put H plus in brackets like that, that little designation means concentration of these hydrogen ions. That's what those little brackets mean. If you have to take a chemistry class, you'll see that a lot more. All right, so if our concentration of hydrogen ions is high, do you guys remember what that means about the pH? Now you might think, oh, well, if the concentration of hydrogen ions is high, then my pH value is going to be high. That's not true. pH will be low. If your concentration of hydrogen ions is low, then your pH number is high. There's a mathematical reason for that. That's beyond the scope of this class. You'll learn about that if you ever have to take a chemistry class. So for now, you just have to know that this is the case. All right, and then if your pH is low, is that acidic or alkaline? If your pH is high, is that acidic or alkaline? Okay, so low pH is acidic, high pH is alkaline or basic. So having reviewed over that, all of that, what's our answer over here? You got a very high concentration of hydrogen ions, so that lets us know we have a low pH, and low pH solutions are acidic. So acidic, and we have a low pH. Answer there will be D. What's neutral on the pH scale? 7.0. Human body fluids, we said normal pH range for human body fluids, 7.35 to 7.45. And that's not neutral on the pH scale. That's a little bit on the basic or the alkaline side. Our body fluids are a little bit alkaline. Okay, some more terminology, chemical reaction terminology here. Here's an example question. Anabolic reactions. You have metabolism is a term that refers to all of the chemical reactions of the body, and that consists of catabolism, if you guys remember, plus anabolism. And that's because most chemical reactions are either building chemical reactions or they're breakdown reactions. You're either taking two smaller molecules and putting them together, or you're taking a larger molecule and breaking it apart. So a good way to remember that, anabolic, like anabolic steroids, like a bodybuilder would take, the anabolic reactions are building reactions. So like when you build your muscles, you've got lots of anabolism taking place. Those types of reactions then involve a lot of synthesis. Catabolism is the opposite. So you've got a lot of breakdown or decomposition that takes place with those. Then the other thing you have to remember is, in general, when you take a bigger molecule and split it into two smaller molecules, that releases energy. There's actually energy stored in that chemical bond. So when you break the bond, energy gets released. And on the flip side, whenever you do a building reaction, a synthesis reaction, you're taking two smaller molecules and putting them together, having to force them together. It takes energy to make that happen. And so these building reactions require energy input. So having reviewed all that, how would we answer this question? Anabolic reactions, synthesis, and since we're building things, it's going to require energy input. So our answer there is C. All right, a little bit more chemical terminology. The types of reactions that bond organic monomers, like monosaccharides, together to form organic polymers, these are chains, larger molecules, are generally which type? Which type of chemical reaction So remember, here we're thinking about anabolic type reactions, synthesis, building type reactions. We're going to have energy input to make that happen. And if you go back and review these, like when you're taking amino acids and linking them together to build proteins, those types of reactions are a dehydration synthesis. And the reason for that is if you've got here's monomer number 1 over here, and we'll have a hydrogen there, and monomer number 2 over here with an oxygen and a hydrogen attached there. When these get linked together, those go away, and 1 and 2 wind up attached to each other, and the H from there and the OH from over here form a water molecule. So it's dehydration because the two monomers together have lost a water. Okay, organic compound review here. The following monomers can be bonded together in chains by what kind of chemical reactions? What do you do to build larger molecules from smaller ones? So we just went over that. That's dehydration, synthesis, chemical reactions. These reactions are anabolic or catabolic. Anabolic, because we are taking two smaller molecules and sticking them together to build something larger. Does that require energy input or output? Input. You got to have energy input to make that happen. Just like if you're building a house, if you take bricks and put them together in an organized fashion to build a wall, it takes energy input, work, effort to make that happen. All right, what organic macromolecules are built from chains of each of these monomers? So this is just information you got to learn. What are the building blocks of these organic macromolecules? Amino acids, you chain those together, you make a protein or a polypeptide. Polypeptide is another name for a chain of amino acids. Monosaccharides get chained together to form polysaccharides. Carbohydrates are built from monosaccharides. Nucleotides, those are the building blocks of DNA and RNA, your nucleic acids. Fatty acids and glycerol are the building blocks of lipids. So be sure you go back and review that terminology. That's basic chemistry stuff that comes up over and over and over again in your biology courses. So you'll see that in every class. Okay, when you consume carbohydrates in your foods, the digestive system has to break them down into these absorbable monomers. So when you guys take Biology 202, you're going to learn about the digestive system. And like if you're eating something that's built from complex carbohydrates, you've probably heard before about you need more complex carbohydrates in your diet. Before, carbohydrates are built from monosaccharides or sugars. Well, you can't absorb a large carbohydrate molecule through your intestinal tract and into your bloodstream. You've got to chop that carbohydrate into its monomers, and then those monomers are small enough to actually be taken across the lining of your small intestine so that you can absorb them into the blood. All right, so if you take a carbohydrate and you chop it up into its individual building blocks, what are those building blocks? All right, hopefully you answered sugars or monosaccharides, another name for sugars. And we know it's not amino acids because where would you get amino acids from in your diet? From breaking down proteins. Fatty acids, you get those in your diet from breaking down larger organic molecules that we know as lipids. Nucleotides come from DNA and RNA. Okay, here's one of those pesky select all that are true questions that everybody hates, but here's the approach you want to take with these types of questions. You don't, what you want to do is read each answer and treat it like a true-false question. Don't try to second guess. Don't try to go through these mind game kind of things where it's like, well, I don't think 5 out of these 6 would be correct, so I'm only going to pick 3. Or, you know, don't start heading down that pathway. What you want to do is just go with each one and treat it like a true-false. All right. Select all that are true about proteins. All right. How about A? Are they built from amino acids? Yes, we just went over that more than once. Are they the locations of genes? No, genes are on DNA, not proteins, so that's not a correct answer. There are 20 different kinds of amino acids available in your cells for building proteins. Yes, that's true if you review some of that basic info. Do proteins play many diverse roles in cells, such as functioning as enzymes? Yes, proteins are sometimes called the workhorses of our cells, the chemical workhorses, and almost all enzymes are proteins, different kinds of proteins that our cells make. Are they our main sources of short-term energy? No. So what type of molecule should you be thinking about as short-term energy? Carbohydrates, sugars. You guys know that. If you consume something with sugar in it, that's a good way to get a quick burst of energy. Proteins generally hydrophobic? No, not usually. What kind of organic molecule have we studied that's generally hydrophobic? Those would be your lipids, your fats. Oils are lipids. You guys know oil and water, those two things don't mix. So compounds that hate water and won't dissolve in water are hydrophobic. Most proteins are hydrophilic. They like water and they'll dissolve in water just fine. All right, when you have a DNA molecule, if you guys remember, oops, almost get my answer away there. You got a DNA molecule, you got 2 strands of DNA. If you have a C over here, a cytosine on one side, what is that going to pair up with over on the other side? G, a guanine. So our answer there will be C, guanine. If you had an A over here on DNA, what is that going to pair up with over on the other side? T for thymine. A stands for adenine. Uracil, that answer that you see there, remember you only have uracils in RNA, and you have those in RNA in place of your thymines or your Ts. An amino acid would not be an answer there because amino acids are the building blocks of proteins and not DNA or RNA. Okay, be sure to review the things that we've learned about cell membranes. Which of the following is not true about the plasma membranes of your human cells? So let's just go through these as well. Again, you can pause and try to answer before I start discussing the answers. How about A? Are they mainly composed of phospholipids and proteins? Yes, that's true. You've got remember a cell membrane has a phospholipid bilayer, but then there are lots of, got that bilayer there, but there are lots of proteins sitting in the membrane that have various functions that we'll hear about more in both of your A&P courses. Do they contain cholesterol molecules? Yes, they do. They're kind of scattered around. They're smaller lipid molecules that have a little bit of a different chemical structure, but they're really important for your membranes to have proper function. So we do have cholesterol molecules in our cell membranes. Are your cell membranes selectively permeable? Yes, they are. Hopefully you answered yes to that. That means that they let some substances across, but not others. So they're selective or picky about what they allow to move across. So that means D obviously is our incorrect answer. That's not true. Oh, I'm sorry. it's the answer. It's the correct answer, but it's the one thing that's not true about your plasma membranes. And that says they have hydrophobic surfaces facing intracellular and extracellular fluids. No, that would not be correct. Your intracellular and your extracellular fluids are water-based, so you're not going to have a hydrophobic substance facing those fluids. You're going to need a hydrophilic substance facing those fluids. Okay, review what we discussed about things being transported across cell membranes. This is a topic that's going to wind up being important a little bit later in the semester. Which of the following is true about diffusion? I'll let you guys answer that. Let's go through these. A, diffusion only applies to water molecules when thinking about cell membranes. No, not true. Anytime we've got a substance that is moving from an area where it's more concentrated to less concentrated, that's diffusion. So water does that, but so do lots and lots and lots of other things as well. So it doesn't just apply to water. There is a special term, though, when we're thinking about the diffusion of water across a cell membrane, and hopefully you know that that term is the O word, osmosis. Very important topic that's going to come up a lot in your 201 and 202 courses. All right, how about B? Substances tend to move down a concentration gradient. That's true. A concentration gradient means that substances are moving from where they're more concentrated to less. In reality, this movement is random, so we talked a little bit about that. So like imagine if you had a beaker of water and you put in a blob of ink into that water, and those ink molecules and the water molecules in the beaker, they're all just kind of wiggling and jiggling around randomly, and eventually everything's going to even out inside that beaker. You're just going to have kind of a uniform mixture of the water and the ink molecules. But that's diffusion. It's just this random motion of molecules that tends to even things out. But to our eye, it looks like the ink moved from where it was more concentrated to less. And that's what we refer to as a concentration gradient. All right, so we know C is wrong. Diffusion occurs because molecules and ions are very rigid structures that have no motion. No, molecules and ions are always randomly in motion, kind of wiggling and jiggling around. And D, substances move from where they're less concentrated to more concentrated. That's not true. That is the reverse of what's true. All right, how about this one? Which of the following would you expect to be able to diffuse directly through a phospholipid bilayer with no special assistance? All right, so to answer this, and no special assistance means, okay, so if you've got a cell membrane here, cell membranes have lots of things like channels and carriers that can pick up substances on one side and dump them over on the other, or channels that will allow substances to move from one side to another. That's what's meant by assistance. You know, if you're using some sort of protein to help you get across the cell membrane. So there are some substances, though, can just diffuse from one side of the membrane to the other, right through the phospholipids. And so which of these could do that? Let's think about this. Sugars, proteins, ATP, or a hydrophobic compound? All right, hopefully you picked hydrophobic compound because you know the phospholipid bilayer is hydrophobic. The inside of that is hydrophobic. Hydrophobic substances like other hydrophobic substances. So that's the only thing that can actually worm its way efficiently, or one of the few things, the only thing among these that we see here in this question that can accomplish that. Sugars are hydrophilic, so they're going to stay on one side of the membrane or the other unless there's a protein in the membrane that can help transport them across. Proteins are big organic molecules. Really big molecules don't freely move across cell membranes. And ATP, our fuel molecule, our energy molecule, is a nucleotide, which is also a hydrophilic type of molecule. So it's water-loving, and those types of molecules don't just freely diffuse across cell membranes. The only things that diffuse freely across cell membranes are some types of hydrophobic compounds and then little bitty tiny inorganic molecules that don't have a charge. Things that have a charge love water. You guys know that, like electricity loves water, so think about it that way. But smaller molecules like oxygen gas, CO2, those types of things can also freely move across cell membranes. All right, here's another transport question. If you had a monosaccharide and it was more concentrated, we've got a cell membrane, and we'll say this is inside the cell, we'll say this is outside, monosaccharide, 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 and it's more concentrated outside than inside. How would we expect it to enter the cell? So let's think about this. It's more concentrated outside than inside. Which way is it going to naturally want to flow? So we have to think about diffusion. Things move from where they're more concentrated to less. So we know that it wants to do this. It wants to move across the cell membrane. Then we have to think about, okay, it's a monosaccharide. That's hydrophilic, so it's water-loving, which means it can't just freely move through the phospholipid bilayer cell membrane. It can't do that on its own. So it's going to have assistance in order to do that. So which of these answers takes those things into account? All right, it looks to me like this is going to be our answer: facilitated diffusion. It wants to diffuse naturally from outside to inside, but it needs some help. It needs diffusion to be facilitated or helped out, and that occurs through a carrier or a channel protein of some type. Active transport, if you go back and review, sometimes cells need to take up substances or get rid of substances against their concentration gradients, which means they need to move from where they're less concentrated to more, so it's an unnatural method of movement, and that's where active transport comes in. And that involves pumping, usually through a carrier protein of some type. The cell uses energy to force a substance to move in the wrong direction, basically. So simple diffusion, that can't occur because the monosaccharide is hydrophilic. Phagocytosis, if you guys remember, that is a process by which like a large particle binds at the surface of the cell and gets taken in, engulfed inside a little membrane-covered vesicle or compartment. So that's generally for larger substances. Like you have white blood cells that do phagocytosis to help them eat and digest bacteria that you get exposed to. That's an example of phagocytosis. All right, here's another transport question. Suppose sodium ions are more concentrated in extracellular fluids than intracellular fluids. All right, so we got our cell membrane again. Outside, inside. More concentrated out here. Less concentrated in here. But suppose these sodium ions need to exit anyway. This is going to come up when you guys study the muscular and nervous systems later in the semester. How do we get those sodium ions out? So you can't just open up a channel. If you just opened up a channel, which way are the sodiums going to move? They're just going to flow that way if you just open up a channel. Instead, we need to deliberately force these sodium ions to go this way, out of the cell, and like we just mentioned, that involves active transport. That's where a cell uses energy and some sort of like a carrier protein to force things out of the cell or into the cell against the direction they naturally want to go. All right, osmosis. Super critical topic. Be sure you review osmosis and how that works. All right, so we got a cell here. I'll draw our little cell. And ECF stands for extracellular fluid. ICF, intracellular fluid, the fluid inside the cell. Extracellular means outside the cell. And all right, so it says there's a higher concentration of solutes. What are solutes? Be sure you review that. Solutes are just any of the dissolved substances in our fluids. So it includes things like ions, glucose, amino acid, proteins, any of those things that are dissolved in the fluid. If you have a higher concentration of those inside the cell and a lower concentration out here, what is true about the ECF? How do we classify the ECF? This gets into the tonicity or tonic terminology. So if your solute concentration is lower in the ECF than it is in the ICF, what prefix there means less? Hypo. So our ECF is hypotonic to the ICF. We would say that our cells are surrounded by a hypotonic fluid. Hyper means greater or more. The ECF, how about this one? The ECF has a lower osmolarity than the ICF. Okay, that's also true because this term osmolarity just refers to solute concentration. That's all that's referring to. So if we have a lower concentration, oh, never mind, guys. I'm talking about this in reverse. Hang on. Let me start. I can't read. You guys are probably sitting there thinking I'm insane. ICF, ECF. See what I mean about having to read questions carefully, and I'm sitting here yakking away, not even reading the question carefully. The ECF solute concentration is higher than it is in the ICF. So your ECF is hypertonic. I'm starting to wonder why there was more than one correct answer here. All right, so it's not hypotonic. And it does not have a lower osmolarity because that would mean there would be a lower concentration of solutes in our ECF than in our ICF. And here we see that we have a higher concentration. The ECF is not isotonic. What does isotonic mean? Isotonic would mean the solute concentrations were the same on either side of the cell membrane because iso means same. That's what that prefix means. So be sure you learn iso, hyper, and hypo, what those prefixes of these words refer to. Sorry about that. Sitting here telling you guys to read questions carefully, and I'm doing the same thing I'm telling you guys not to do. All right, Louis guzzles many gallons of pure distilled water that contains no solutes, but refuses to urinate excess water. So he's not allowing himself to go pee out this excess water. What's going to happen to his ECF? All right, so if you're guzzling lots and lots and lots and lots of water, not letting yourself pee out the excess, here's our ICF, here's our ECF. That water, remember, fluids move from compartment to compartment inside the body. And so if you're guzzling all this distilled water, what's going to happen to your ECF? What's going to happen to the solute concentration? It's going to get diluted. So your solute concentration will drop in your ECF. Okay. And so how are we going to refer to your ECF in this case? Is it going to be hypertonic? Is it isotonic? Is it hypotonic? Okay, and hopefully you are choosing, remember, solute concentration is going down because we have more water. Everything's more diluted out. So his ECF is becoming hypotonic. Hopefully his bladder won't explode. Now, you guys may have heard before of water drinking contests where people have actually died, or sometimes fraternities have done hazing like this where they have people drinking lots and lots and lots of water, tons of water, and not allowing urination so that you can get rid of the excess water, and it can actually kill you because if your ECF becomes very, very hypotonic, what's going to happen to the cells, like up here in your brain doesn't have much room to expand, so your brain tissues take up this excess water, the brain swells, and that results in death in some cases, or at a minimum, an extremely painful headache and becoming disoriented. And that doesn't mean you have to fear water. These are extreme cases where you're taking in tons and tons and tons and tons of water. You're not taking in any solutes, and it winds up causing your ECF to become hypotonic enough to make your brain swell. It's called water poisoning. Sometimes people who do marathons or triathlons, like if they go to fluid stations and they just drink a lot of water, they don't drink Gatorade and replenish their solutes, and there are cases where they can get water poisoning as well if their ECF gets diluted out. Those are unusual cases though, so I'm not telling you to avoid water. Water is a good thing for you to drink. Just don't drink gallons and gallons of it and don't let yourself pee. That wouldn't be a good thing. All right, another osmosis question. You can tell I like these osmosis questions. What would happen to your cells if you injected high volumes of very salty fluids directly into your blood? All right, so ICF and ECF, let's think about this. You put a bunch of very salty fluids directly into your blood. So what's going to happen? Blood fluids are a type of ECF. What's going to happen to the solute concentration of your ECF? It's going to go up. It's going to become very salty. The osmolarity is up. It has become hypertonic. Now let's think about water movement across the cell membrane. Remember, these solutes, like we were just talking about with the cell membrane, most of these solutes, like ions and glucose and amino acids, all that stuff, those things don't freely move across cell membranes. Water can, though. Water is a small inorganic molecule, and it's able to wiggle its way through the cell membrane, even though that sounds counterintuitive to what we've been studying about the cell membrane. Water molecules are so small that they can move across. And water, remember, water is attracted to solutes. All these hydrophilic solutes like glucose and ions and so forth, they love water. So if you have more water out here, if you have more solutes out here, water molecules that are inside the cell are going to diffuse towards those solutes. They're attracted to the solutes. Water follows solutes. Water follows salt. And so what's going to happen to your cells? Are they going to shrivel? Are they going to swell? Are they going to remain unchanged? Are they going to crawl around in your tissues gasping for water? Which of these will happen? Well, we just said the water molecules are going to move out of your cells, so hopefully you have chosen that they would shrivel. What if and like we said, if you had a case of water poisoning where your ECF, the solutes were very diluted out, so you got tons and tons of water molecules in your ECF, and you've got more solutes inside your cells, the fluid inside your cells is hypertonic. In that case, water is going to move from the ECF to the ICF. The water is going to be attracted to those solutes inside your cells. That causes your cells to swell rather than shrivel. And your brain doesn't have much room to expand. So when the cells in your brain take up a lot of water, the brain presses up against the cranial cavity. There's a big hole in the bottom of the skull for the spinal cord, and parts of the brain stem, get squeezed through that hole, and that's what can actually cause death to occur. All right, so in Chapter 3, we talked a little bit about organelles. These are parts of your cells that have specific functions, so just review the basic information about those. Which organelle is responsible for producing most of the ATP fuel that's needed by our cells? And hopefully you picked mitochondrion. Mitochondrion is singular. Mitochondria is more typically, with an A on the end, is more typically how you see that word, and that's the plural form, because generally cells are going to have more than one mitochondrion because those are your powerhouses. So review the basic information about the cell organelles. We didn't spend a tremendous amount of time. Oh, I skipped over to the next question and didn't even realize it and gave away the answer. Which organelle packages proteins into vesicles and directs them to other locations such as the plasma membrane? And the answer there is your Golgi apparatus. Some people refer to that as the post office of a cell. So you've got this, a lot of proteins that are made in cells are traveling either to the cell membrane or they're going to be released, exported to the outside of the cell, and they go through an assembly line process where they go from the rough endoplasmic reticulum and then travel in vesicles to the Golgi apparatus, and then from the Golgi apparatus they go to the cell membrane or other organelles, or they get secreted, released outside of the cell, and that sort of decision making occurs at the Golgi apparatus. Your Golgi is kind of like your cell post office. All right, so those were just some example questions I wanted to go through. That does not mean that those are test questions or that those are the only things you will be asked about on the test. I just wanted to kind of go through some example questions and talk our way through how to approach those questions and think through the material that you've been learning. And again, like I showed you, I wasn't reading carefully and I was picking the wrong answers on a question. So you got to be real careful with reading your questions and rereading them and making sure you're not making a mistake like I just did. So I'm not even going to edit the video and edit that out. I'm going to leave that in there because it turned out to be a really good example. Good luck with your studying. Please post any questions you have over on the Piazza discussion boards, because again, yes, I don't mind you guys sending me course messages with questions that you're having, but it would be better if you'd put them over on Piazza, because if you have a question, don't feel like it's a dumb question. Nobody has sent me a dumb question about the material yet. I don't consider any questions dumb. Well, okay, maybe I could at some point if you were asking me about Mickey Mouse or something crazy like that, but if you're asking a question about the material, in all likelihood, another student has the same question. So you can post it on Piazza, and then we'll have our little discussion about it, and everybody can benefit from it. And if you're still too bashful, Piazza has an option where you can post anonymously. So nobody will even know who's asking the question if you're concerned about it. Okay, well, good luck with preparing for the Unit 1 Physiology exam. And remember, everything that we just went over applies to that test rather than the anatomy test. The anatomy is just practicing with images and pictures and practicing your spelling and your identification, and you can do that with the practice activity in the Unit 1 anatomy folder. Good luck, and hope everybody does well on the Unit 1 tests. And as soon as you're finished with it, please go ahead and get started on Unit 2. Things tend to move very quickly during the semester. Take care.